Hi, my name is Brett Goldsmith. I'm the CTO and co-founder of Cardea, and today I'm going to talk to you about what Cardea is doing to help change how the world conducts biology. So we think about biology in a similar way to how we think about all the other technology that's out there in the world. And when we think about technology, the thing that comes up the most is the power of digital networks. Digital networks are critical to how we live today, particularly right now in the middle of the COVID-19 crisis. They're how we communicate instantly and globally around the world. They have these properties that their form is fractal. You can zoom in and you can see that there's complexity at the local scale. Your local computer has a network, and at the global scale, the internet has a network, and those networks have the same sort of topology. It's an engineered, human-readable, scale-invariant topology, and it's super powerful. Now, biological networks have some interesting similarities to digital networks. They're also scale invariant. So if you zoom into how a cell works, it's going to look similarly in terms of the math that describes it to how an entire ecosystem works. But unlike digital networks, biological networks are self-organized, and they're not engineered. They're not human readable. They're not. We're not capable of just looking at them and picking out this gate here is engineered this way, and this little bit of information there is designed to flow this direction. They're just incredibly complex. They're also dynamic. So digital networks do change, but they change on purpose. We're engineering the changes over time. Biological networks are dynamic. Everything about them, the structure of the network and the function of the network, are linked. Everything's changing all the time. And the last really important feature of biological networks that distinguishes them from digital networks is their hyperlocality. There's no global communication of instantness with biological networks. Every piece of information has to touch another piece of information in order to be transmitted. And as we can see with the global pandemic, that can happen, and it can happen on a shockingly fast timescale. But there remains this need for a chemical interaction in order to pass information from one place to another. What we envision at Cardea is taking a technology that we call biology-gated transistors and connecting these worlds of digital networks and biological networks. And so on the right here, you can see a couple of different examples of what we're getting from the electronics side of the world: wireless networking, personal devices, electronics fabrication, one of the most significant manufacturing advances that humanity has ever seen. Machine learning, an ability to start growing our understanding beyond what a person can, or any collection of people can, by leveraging software in ways that seems, illogical but extremely functional. On the biology side, we're bringing things that the hardware and the electronics cannot match. Things like genomics, microbiomics, synthetic biology, and proteomics, an ability to engineer highly complex things, not in the same way that is done on the electronic side, where you're building bottom up, but where you kind of guide, you evolve. And so we've represented that here. The biology side's got these curves, and it's kind of messy. But biology works. We're alive. We know it's functional. And we're starting to tap into the power of understanding it through the genomics and proteomics and microbiome side. But to work with something, to engineer it, to manufacture it, to make it accessible to everyone in the world, that's something electronics has done. You can find cell phones everywhere in the world. It's amazing. But you can't find ELISA kits. You can't find PCR machines, unfortunately, everywhere. So our biology-gated transistors are the key, the gateway that's going to connect the biological world, the messy, complex world of biology, to the engineered, global world of electronics. And we say that this begins the era of connected biology, the era when the local, the hyperlocal biological networks can be combined seamlessly with the global digital networks. And this will allow us to do things like Google, what is my cholesterol? And have that response come up as quickly as asking, what is the capital of Kansas? So this is what we envision. This is our vision for the world, for the future, that we're enabling an infrastructure around biology-gated transistors that's going to directly access biological information in real time. We're not talking about taking data. We're talking about taking data streams. And this is going to transform the way we conduct biology. And that's a pun. We're making electronics and we've got conductors. So we're conducting biology. But it's also about how we change the action of biology from an engineering point of view. So, okay, those are really grand claims, and it's a really bold vision, and it's the kind of thing that you might have heard circling around nanotechnology conferences for the last 20 years. And a big question has always been, well, how do you do this? Is this realistic? So the answer, of course, is yes, and let me explain to you how we do it. So we do something, again, we've called it a biology-gated transistor. It's a graphene transistor. It's not a digital transistor. It's not a transistor in the same sense that your computer runs on. It's more like a transistor from the radio days. We put a voltage on the gate, and the gate changes the resistance. It doesn't go on and off, but we see a change in the resistance with the change in gate. And in our case, the gate is biology. So here on the left, you've got our transistor where we've placed our biological capture mechanism. This could be any sort of active biology, an antibody, a piece of a virus, a piece of a cell, a piece of DNA. And we're looking at on the right side, the analytes, the things that biology interacts with. They bind or they don't bind. And if something binds and the surface changes and that gate changes, then our little dial, the base signal, goes from white to yellow. We see a change in the current through the transistor. And I'm going to get into some more details on that just now. So this is a little bit more detail here. So one thing that happens, so we're using graphene. Graphene is a new electronic material. It's different from silicon. It's different from any of the traditional semiconductors or even the metals that you're used to seeing in electronics. It doesn't have surface oxides. Basically, what that means is chemically, it doesn't oxidize. It doesn't rust in air. It doesn't rust in salt water. That's unique. You can take silicon and you put it in air, and it's going to create an oxide. You take any semiconductor, you put it in air, it's going to create an oxide. That oxide is a chemical barrier between your electronic part and the rest of the world. Most electronics just does not live in the world we live in. It doesn't coexist with biology. Graphene does. So you can take the graphene and you can attach the biology to it without anything in between. Now, we do use a little linker molecule to make sure we get the right biology in the right place. But this is a far different scale than an oxide. The linker molecule and the biology that we attach are what give our sensors their specificity. So if we want to look for a specific antigen in your blood, we have to attach the right antibody. And if that antibody is good, we're going to have a very sensitive sensor. And if that antibody is crummy, we're going to have a crummy sensor. The graphene allows us to detect the changes happening in the biology with extraordinary sensitivity, but it doesn't do anything magic to the biology. If the biology itself is not sensitive, then the sensor as a whole is not sensitive. This is a technology that is a combination of biological technology and electronics technology. So you need both to function well. Here you see we're a couple little more diagrams about how the sensor is working. We've got two electrodes that are controlling the gate. So we have kind of a little electrochemical gate here. We're encapsulating the electronics. There's a lot of layers that go into this, into the manufacture of this. But for a silicon chip, for a piece of manufactured electronics today, this is a very simple design. That said, it's one of the more complex pieces of nanotechnology that are out there. So we've been at this for a little while. In nanotechnology, we've been making devices like this for about 20 years, a little more than 10 years, maybe 15 years that we've been making graphene devices. But commercially, we started in 2013 in my garage, making the device you see on the far left there labeled Alpha. It's the kind of thing you'd make in your garage. We went from there, and the cost for that one device was astronomical when you really think about it. It took months to make one device, and that is typical of the kind of handmade, artisanal nanotechnology devices that have been made in the past. The kinds of things that I made during my PhD project and that PhD students are making all the time right now. Often we get a discount on the real price because we're trading academic achievement for salary. But if we're going to commercialize something, someone needs to make it and they need to get paid a reasonable wage. And when you project things forward, it just doesn't work out unless you can get into a fab. So we go from the alpha to the first prototype, starting to see how can we get this onto a printed circuit board and something that is a little bit more scalable. We get to what is called our beta chip. Now we have something that is actually manufactured on a wafer scale. It's professionally packaged, and chip packaging is one of those things that it is extraordinarily difficult, extraordinarily expensive. So early forays for us into commercial packaging, and then finally we get into our first commercial chip, what we called at the time the test tab. Now these are commercial chips that are sold. We've had customers, we've had lots of successful customers that have done their own research, published papers, starting with that green first commercial chip that you see right there. And over several iterations from that, we went from the first chip to the second chip to what we call the first Cardea chip. And what we mean by that is the first chip that we really feel we can make at scale with a level of reproducibility and quality control that we're ready to partner with people on large-scale projects and integration into larger systems. It has a lot to do with cost, the real cost. At the end of the day, after all the overhead and everything else and quality and the shipping and everything is put together, moving from back in 2013, a real cost of hundreds of thousands of dollars per chip to today costs of tens of dollars per chip. And that just opens up a whole new world of what can be done with these kinds of devices, this kind of nanotechnology. So I wanted to give you guys an example of the kinds of things that we do with these sensors. And so this describes one of our early projects with the CDC. The CDC was one of our earliest partners. They were around for some of those really early chips that didn't look so good. And they were big believers in the vision, big believers in the idea of having something that was electronic and fast and portable. And you can imagine why. And so this particular work, we took some antibodies against Zika that our CDC collaborator had developed, and we attached them to our chip. And then we put in different types of blood samples. Now, I didn't have actual Zika-infected blood in my lab because it's not set up for that. But we were able to take clean blood and spike in proteins from Zika at a level that would be the same that you'd see in actual infected blood. And we'd compare the clean blood to the blood that had the Zika virus protein spiked into it. And when we did that, we were able to see clearly that the Zika virus infected blood gave a much different signal than the healthy blood. And you can see that here in this graph in the middle, where the really cool thing here is the time axis, that x-axis there. So we spent 5 minutes just calibrating in a clean sample. And then we inject either a different clean sample or the infected blood. And you can see that although we took the measurement for 15 minutes, it took just a couple of minutes before we could clearly see that there was a huge difference between the Zika protein spiked blood and the healthy blood. Now, this was all done on a system you can see here on the right that is connected by USB to a laptop on a reader, and that's an actual picture. That's someone's hand there. It's not a very big thing. It's something that's handheld, something that could be portable. And so you could see that this is the first effort that we were making to try and show people that what we're doing could have real impacts for the world of finding and diagnosing and monitoring infectious diseases. Because if you can get something like this out into the field, where you can do an assay in 5 or 10 minutes to detect presence or absence of an infectious disease, that can make a big difference. So another thing that we did early on is we started looking at some common inflammation markers. And inflammation markers like IL-6 here. These are the kinds of things that happen when your immune system is ramping up, when you're trying to fight off something. And it could be an allergy, could be a disease. Now, overexpression of some of these markers is really a big problem with, for example, people who have COVID-19. And so one of the things we were trying to do here, this was before COVID-19. We were simply trying to show that we could detect these inflammation markers faster than you could with the standard ELISA kit, and that we could meet the same concentration requirements. We get the same limit of detection. And the reason why you want to do that is if someone is having an immune response or an allergic reaction or starting to get sick, you don't want to wait around hours for a labeled test for some evolution of a dye or to get onto a plate reader. You just want to know right now. And so if we can do that electronically, locally, wherever the patient is, then that would be great. And so what we have here on the left is the data from an IL-6 ELISA, and that's the standard technology that's used to do these kinds of tests. And this is the optical absorbance, and this is taken on a plate reader with lots of different concentrations, log scale on the x-axis. And the limit of detection for this we saw was about, let's see, I can't read it, 68 picograms per milliliter, pretty good. So we did the same thing. Now we did this in cell growth media, so it's a bit harder than just doing things on a buffer or in water. This is something that has a lot of different stuff in it. It's something that cells were growing in. It has different pieces of cells and all sorts of stuff in there. And we did this to simulate a real world environment. One of the places you might find IL-6 is in cell growth if you're trying to grow cells for a particular purpose, and you're worried about stressing them out. And so the limit of detection that we see is 132 picograms per milliliter. Now that's higher than what we saw in ELISA, but the difference is that one of these is an endpoint assay. It's a test that you're doing. You're extracting some material, you're sending it off to a lab bench and running it through a gamut of different wash steps and dye measurements until you get your result. Ours is a real-time measurement. We're getting those results in real time. You're not waiting. You can stick the sensor right in the media and read out the data right then. So it's quite helpful to be able to get these kind of very fast, very sensitive measurements. And there's more details on this kind of measurement in another paper that we published last year in Scientific Reports that shows that when we're operating in buffer, we actually were more sensitive than the ELISA assay. The last example that I wanted to show you was detection of leptin in blood. And this is something that was done with some early collaboration at UC Berkeley and also Keck Graduate Institute. Now, in this study, we were detecting leptin as part of a study involving aging. So this was just part of the aging study. And this was the kind of thing they had a little flow-through sensor that they built on top, or a flow-through channel they were built on top of our device. They were extracting blood, and they were looking for these aging markers. They took the leptin itself, and they put the leptin down onto the chip, and then they were actually looking not for the leptin, but for the leptin antibody in the blood. And they were trying to track the concentration of that antibody in the blood as part of the study. And so you can both take the chip and use it to detect a particular antigen like IL-6 or the Zika virus protein, or you can flip it around. You can put the antigen down and you can look for the antibody against it. In that way, you can see that you could look for something like, have you developed an immune response to something? You could imagine, hey, have I had COVID-19? Have I had this strain of COVID-19? Have I had this year's flu yet? Those kinds of things could be answered with this kind of test. That was also published 2018 in Lab on a Chip. Last example here, and this is the one that probably has gotten the most attention for what we've done, and that is now the most biologically complex measurement that we've done, and that's CRISPR on the chip. So CRISPR is a combination of proteins and RNA that are used together to detect DNA. And we did this also in collaboration with UC Berkeley and Keck Graduate Institute. We did detection of DNA, specific detection of DNA without amplification. And that's something that's really important because amplification causes a lot of errors and can cause a lot of problems. It's one of the big issues with how do you identify a rapidly mutating virus, for example. Sometimes the selection of the amplification probe can really determine the efficacy of your test. Now, we have an ability to do multiplexing of lots of different tests across one chip. We can look at lots of different DNA strands simultaneously. There's not a necessity for us to be limited to one probe or a few probes. We're limited only by the aggressiveness in manufacturing that we pursue. The electronics industry, the chips in your computer, they have a lot of transistors on them. We can pack a lot of transistors and a lot of sensors onto a chip. So let's talk about this specific assay. We take the Cas9 protein. That's part of the CRISPR complex. We immobilize that onto the chip. Then we take a search query in the form of an RNA sequence. And that search query, RNA sequence, is then loaded into the Cas9 protein, which is already on our chip. And that forms a CRISPR complex. Now, in this paper, we used dCas9, meaning that it was Cas9 that did not cut the DNA after it found it. It just searched for the DNA. If it found the DNA that we had coded it to find, it would lock down and stop, and just hold it in place. And what we'd do is we'd wash over genomic DNA, all the DNA in a sample, everything. Let this little biological machine search through that DNA for the match. And if it found the match, it would lock it down. Then we'd rinse off all the DNA that wasn't bound to the chip. And if the DNA that we were searching for was found, we would get a big response. And if the DNA we were searching for was not found, we would get a small response. This searching took about an hour. So sometimes we could go as low as half an hour, but about an hour to do this entire search operation. If we're trying to do that computationally, if we're trying to do that via electronics and software, it takes a lot longer to search an entire genome than an hour when you're talking about sequencing or something like that. So this is not a sequencing technology, it's a DNA detection technology, but it does DNA detection faster than any combination of other electronics that we can find. And it's because we're leveraging the power of this biological machine, the CRISPR complex. And this all makes sense. The CRISPR complex is a bacteria's immune system. Its purpose is to rapidly search out DNA that might be part of a virus and to cut it up. So if you're looking for DNA that's part of a virus, doesn't it make sense to use the biological machines that do that? So here on the right, you can see the cover of Nature Biomedical Engineering, a paper that all of this data is from, and then the key piece of data from that paper, which is we were able to take some samples that were both healthy and not healthy for Duchenne muscular dystrophy. And Duchenne muscular dystrophy is a lack of a gene in a particular set of, or a lack of an exon in a particular gene. So a particular set of DNA is missing. And what you can see here in these graphs, all these bar graphs, the gray graphs are either healthy samples or samples that are known to be missing a different gene than the particular one we were looking for here. And we can see that in all of those samples, we find the entire gene, all the exons that we were looking for. But in those blue samples, those are the ones that were missing exon 51 out of this gene. And we see that those samples give us a lower result, meaning the DNA did not stick to the surface. It was not locked down by the CRISPR complex when it did its little search mechanism. So these four examples give you an idea of the breadth of sensing that we can do, the different kinds of things where we can take antibodies, we can search for viral proteins, we can search for immune responses like IL-6, we can search for antibodies that your immune system has generated against proteins, and we can search directly for DNA through the CRISPR complex. So something that's kind of interesting. Now, switching gears and talking a bit about COVID, when you have a patient and you're looking at viral load and whether you have symptoms, is it sufficient to just say how much virus do I have on me to know whether or not, is it sufficient to say am I symptomatic? Do I feel bad or do I feel healthy? Yeah, because you could feel perfectly healthy and still be out there infecting people. So wouldn't it be nice to be able to know that you're shedding virus, you're shedding COVID-19, and so you can see here in this graph, whether you're symptomatic or asymptomatic, you've got the same amount of virus coming out. So a test that was able to detect rapidly and kind of in your hands whether or not you're shedding virus would be invaluable to knowing whether you should stay home or whether you should now be allowed to go outside and interact with the rest of the world. Similarly, we talked a little bit about this on the IL-6 slide. The inflammation response. I think this is from SARS-1. The inflammation response in at-risk people, in this case aged versus young people, is heightened incredibly when you're infected. Again, this is with SARS-CoV-1. But we would expect the same thing with the current pandemic. They're related diseases. So you can imagine a test that looked for what is the ratio of IL-6 to CCL2, or are you producing IL-8 or not? These kinds of ideas, these kinds of tests of looking at your immune response could tell you, are you on the way towards a serious infection, or are you going to have a mild infection? Because it's that really hard immune response that's causing a lot of damage to patients. This would allow doctors to more quickly triage people. If it looks like you're going to have a mild case of the disease, you should just go home and rest. But if you're on track to need a ventilator, to need intensive care, let's get that care early and prevent the worst stages of the disease if possible. So what we're thinking we could do for something like COVID-19, or specifically for COVID-19, should we get the opportunity and the partnerships to do it, would be to improve the diagnostic testing capabilities, both directly detecting the virus, detecting your immune response, detecting your immunity. And we could do this by having a highly reconfigurable device for single or multiple analytes, something that could be available for single use, local portable use, or for high throughput testing, something that is seamlessly transfers the electronic results to the cloud, which really is the only way we can effectively track these kinds of infections. So this is the vision of how our technology could be used in something like this. Now, when you look at the technology here, part of the advantage of using a transistor-based approach, a direct detection approach, a data streaming rather than endpoint approach, is that it is applicable in all of these different areas, whether you're talking about at-home testing, point-of-care testing, molecular testing, immunoassays, or central lab testing. Doing it all on the same platform allows you to streamline your assay development, allows you to streamline data analysis and comparison of apples to apples in different locations and from different communities, different points of view, different users. So the factors that go into allowing us to do this is that we're fast, multi-analyte, multi-form factor kind of technology. This chip can be used anywhere you could put it. It could be used in handheld, it could be used in a robot. It's quantitative. It could be used for biosurveillance, and it's rapidly reconfigurable, meaning you can put down different antibodies, you can put down different proteins, you can program the CRISPR complex with different RNAs. You just have to make the chips and then program them before you use them. This is a picture that we've made, our idea of what multi-analyte testing should look like. You've got a small reader that handles all of the operations of the test, a swab system that uses perhaps saliva, perhaps mucus, and is incorporated into a cartridge that houses the chip. The cartridge goes into the reader and it reads out whatever test that cartridge has been biologically encoded to make. And the whole thing could be communicating with a cell phone via Bluetooth or via Wi-Fi, those kinds of things. Because we're rooted in electronics, those are easy to do. The reconfigurability of the biology, that's the hard part. The multiplexing from, say, 16 elements to thousands of elements, that's hard to do. But that could be done on something this small, this compact, and this easy to use. So some of the things we've talked about doing, using a nasal swab to detect viral RNA, to detect your personal viral load. Are you safe to go out into society or not? We've looked at doing a multiplexed serological assay. What strains of SARS-CoV-2 have you been exposed to, and have you developed immunity to? If you know that you're immune, that means that you can also go out safely, at least for yourself. Now, the kind of things that we're talking about from when it comes to sample type, right? We've talked about blood, we've talked about serum, saliva. We can use all of these different things. These are probably multiple different types of devices. We can do the sample preparation off chip. So there's lots of different ways of doing sample prep. That's not what we're good at. We're good at the chips. And we can do integration of the sample prep into the cartridge if necessary. We talked about all the different form factors. I didn't show them to you. So here, this is an example of it on the left, a 256-plex chip, so something with 256 sensors on it. That's more than we currently use in our commercial chips. In the middle is that picture of the handheld reader that we're envisioning. And on the right is a photograph of a robot that we use right now to do, it's not high throughput in the sense of pharmaceutical testing, but it's high throughput in the terms of how many chips we can get through per day. We can set this up to do up to thousands of chips per day. This whole thing works because we've got one chip, we've got one set of software, we've got one way of analyzing the data. Everything goes to the cloud. Our philosophy here is that you want to separate as much as you can any medical expertise, epidemiological expertise necessary from the local on-the-ground people, because you're not always going to have that available. So if you can get the data up into the cloud and out to the internet as quickly as possible into the hands of the people who can do the most with it, you're going to be doing the most good. So that's the approach for the current disease. The other thing is that as we're doing assay development, as viruses change, as the next pandemic comes, we can mine that data in the cloud to go back into the robot to try out new approaches to the assay, to figure out how could we have improved this assay done over here in another country, and create something we call the Excel solver for system biology, a closed loop of fast electronic sensing and cloud-based data analytics with something that's incorporating machine learning or AI, if you will, to find the best approach to taking data, the best approach to processing and performing the assay. Now, we're not a company that's going to be able to do all of this. We're a company that looks to partners for a big part of it. There's people out there that are better at sample handling, at engineering, at software. And really, we're good at the chips, the nanotechnology. So when we go to partner with people, what you get is you get the chip, you get the chip technology. We're looking for people who can work with us at the small scale, the large scale, who can bring their own technology and allow us to spread it out to the rest of our partners. And we're also looking for people that maybe they just want us to handle everything, and we'll find other partners to get the job done. But there's lots of advantages to working with us. I talked a lot today. This talk was focused on infectious disease in terms of application, but there's a lot of different areas that we look at. And this is one of our business slides. This is not a slide I typically give. This is the universe of addressable market opportunities. We're looking across the board. Everything that we've talked about in the last 20 years in nanotechnology is something that we hope to positively impact in the next few years. And we've already got partners in a lot of these different areas. Not to say that we can't use more in all of them as well. So I hope that through this talk, I've given you some idea of what we're trying to do, how we're trying to approach our responsibility to husband our technology into a form that helps the world with the current crisis, helps the world with the next crisis, and gave you some idea of what it is that you could do to help us out. We think that really biology is technology. We've taken, since 2013, those early ugly chips. We've made a lot of progress on manufacturing, engineering, understanding the design space that we're working within. Now, with the right partners, we can really build an infrastructure that changes the way we all approach biology. We all approach the kinds of challenges that we have literally today. With that, I'd like to thank you.